Hi everyone. So thank you for having me here today to give a talk on click chemistry-based methylated triplex-forming oligonucleotides. So Andrew Kellett mentioned some of this work in his talk, but I'm going to kind of build on this in a bit more detail. So first is just kind of an overview of DNA metallodrugs that Andrew spoke about in a lot of detail. So we have the kind of clinically approved iron bleomycin and platinum chemotherapeutics, but a lot of my research is focused on more kind of artificial metallonucleases or chemical nucleases, which are all kind of based on Sigmund's copper bis-fen. So a lot of research has been done to kind of expand these chemical nucleases, including adding extended intercalating systems like DPPZ, which increase the binding affinity with DNA. And then we also have copper caging systems, so TPMA and DPA. These help to stabilize the copper center, with the less sterically hindered DPA having higher DNA binding affinity. Also then focused on marine alkaloids. So this proteoglycan is able to bind with copper and induce double strand breaks in DNA. And it's also self-activated, so it doesn't require a reductant to reduce copper 2 to copper 1 and initiate the DNA cleavage. We also have dinuclear and trinuclear systems. So Andrew mentioned some of these. So we have copper ODA, which can induce the formation of Z-like DNA. And then BPLC6 and TCPi, which are synthesized using click chemistry. But none of these metallodrugs have any specific DNA targeting. They don't target any specific structure or sequence. And so a lot of research has gone into developing hybrid systems that can target these specific nucleic acid sequences or structures. So these include groove binders like distamycin, spermine, and Huxt, compounds that recognize specific nucleic acid structures, with BQQ, which recognizes triplex DNA, and the actin for G quadruplexes. And then kind of closer to what I'm interested in, which is sequence-specific targeting. So you have triplex-forming oligonucleotides, which can target double-stranded DNA, and this peptide nucleic acid that can target viral RNA. So my talk today is going to focus on triplex-forming oligonucleotides. I know we've heard a bit about already, but TFOs can target sites of the genome that have runs of purines on one strand, so the A's and G's, and then consequently the pyrimidines on the other strand. And that's because when these are bound in the double helix via Watson-Crick bonds, they still have two sites which allow for Hoogsteen base formation with the TFO to form the triple helix. So in this work, we wanted to use a TFO to attach it to a metallodrug to deliver to correct gene sequence, and then initiate sequence-selective DNA damage. And this was done using click chemistry. So we have alkyne modified DNA. It can be clicked with the metallodrugs, and then we get the TFO hybrids. So I've been involved with the synthesis and kind of analysis of a lot of different TFO hybrids, kind of from the first generation. These all target GFP. So you have the copper caging systems, TPMA and DPA, and also cis chemotype platinum drugs. And then more recently, we've been focused on a second generation of TFOs that target the EGFR gene. And these are synthesized using strain-promoted click chemistry. We have the marine alkaloid proteoglycan, and then two platinum DPA derivatives. So first, I'm going to start with kind of the copper caging system, TPMA. This work was done in collaboration with Niccolo Fantoni, who was a PhD student in the group at the same time as I was, and he synthesized a range of different TFOs that are all different lengths. They also have different alkyne modifications, so on the phosphate, 5' end, and internally modified alkyne groups. We initially looked at the thermal melting of these to see which TFOs were the most stable, and it was TFO4, which is 32 nucleotides in length that had the best triplex stability, with all of them melting above 45 degrees. When we clicked them with azide TPMA, this destabilized the triplex, which we weren't that surprised about. But once we added copper, we can see that this stabilization kind of increases again, and they all melted above 44 degrees. Then we were most interested in seeing, could these direct the DNA cleavage to a specific gene target? So this is the 5-azido TPMA hybrid. We've incubated it with target and off-target duplexes. So here's the target, the off-target, we see sequence-selective triplex formation. And then we see the triplex and target duplexes bands been depleted while the off-target remains undamaged. We then compared this to free copper TPMA and a free TFO, so they're not clicked, and we see no significant damages inflicted. So not only does attaching the TPMA to the TFO allow for sequence selectivity, it also increases the DNA damaging propensity by pulling the nuclease into close proximity with the DNA. We're now going to move on to kind of the second copper caging system, which is DPA. And as Andrew kind of showed some of this already in his talk, these were synthesized using an enzymatic approach called SNI-PEX, or single nucleotide incorporation primer extension. And this was the focus of a lot of my PhD research. So the first was to generate a library of click-modified nucleotide triphosphates, and these were then incorporated into my TFO sequence using a DNA polymerase. So you get just one modified base at one specific position in the double-stranded DNA. But for it to function as a TFO, it needed to be single-stranded. And this is where the 5' biotin tag on the end of the template comes in. So it's incubated with strip traveling coated magnetic beads, and then through use of magnet and denaturation step, you can pull off the DPA TFO, and a pure solution is achieved with no further purification required. I analysed these then by page. You can see that it was only a single DPA was included. So it's kind of easier if you start backwards and work across this gel. So lane 4 is the primer on its own. Lane 3 is incorporation of the natural thymine base. Lane 2 is the modified alkyne. And then most importantly in lane 1 is the DPA modified uridine triphosphate. Then wanted to study the cleavage properties of this, and this was similar to what we've seen a few minutes ago for the TPMA assay. So the copper DPA TFO was incubated with target and off-target duplexes. We see the triplex and target duplex bands are depleted, while there's minimal damage to the off-target. This was then quantified using real-time PCR, where we have a target sequence where we get triplex formation, it gets damaged, and then a reduced PCR cycles are achieved, whereas the off-target undergoes no damage. We could plot the fold change in intact DNA concentration, where the target shown in green and the off-target in purple. So here I've compared the solid phase synthesis derivative of the TFO with an enzymatic version, and you can see there's no significant difference between the two TFOs around the target or the off-target. This was then expanded to plasmid DNA. So my TFO in this instance targets GFP. We can see this is the undamaged supercoiled GFP plasmid, and then we see the formation of open circular GFP, which means there's a single strand break been inflicted on the plasmid DNA, whereas the PUC19 off-target remains undamaged. Sequencing then, which Andrew spoke about, shows that the damage can be tuned to a different position within the TFO sequence using different intercalators attached to the TFO. I'm now going to move on to kind of some of the platinum work, and these were of significant interest as they were the first cis chemotype of platinum hybrids. And this was done in collaboration with Dr. Joseph Hennessy, and we synthesized azide derivatives of the 3 worldwide FDA-approved platinum complex, so cisplatin, oxaliplatin, and carboplatin. These were clicked to the TFOs, and then they could form cross-links. So these can be intrastrand GG or AG cross-links or interstrand GG cross-links. We looked initially at did they form stable triplexes, and were they just with the sequence, or did the platinum cross-link with other DNA sequences? We can see it's only the target duplex with which they form triplexes, and these higher order bands are indicative of platinum adducts. But to form kind of more understanding of what the platinum was doing, we then labeled the DNA with different fluorophores, so we could analyze each strand of the DNA independently. So we labeled one strand with Cy5 and the other with FAM. We allowed triplex formation and the platinum adducts to form, and then we denatured them, so we could see only the strand with the platinum adduct remained. And it's this Cy5 labeled strand here for this TFO, which showed that this is the strand with the platinum is cross-linked, while the FAM labeled strand can travel through the gel. I'm now going to move on to kind of some of the more recent work, and this is on this strain-promoted TFO that targets EGFR. So first is the proteosin, which is the marine alkaloid. And this was the first step, was to see that if adding an azide group to the proteosin, did it prevent or did it impact the cleavage activity of the marine alkaloid? We done this first in the presence of sodium L-ascorbate, which is the reducing agent, which reduces copper 2 to copper 1. And we see significant damage at all the different time points with the DNA being completely ablated. But it also maintained its self-activated cleavage ability. So this is with no reductant, where we add copper 2 and the proteosin itself can reduce it to copper 1 and cause the DNA cleavage. And we see at all the time points that we get linear, which is a single strand break, or linear, which is a double strand break, and open circular, which is a single strand break on the plasmid DNA. Next then was to design the TFO that this was going to be clicked onto. And these TFOs target the EGFR gene, so it's within exon 5, as EGFR is overexpressed in many different cancers. This TFO was highly modified to kind of increase its binding affinity. So we swapped all the cytosine bases with methylcytosine, as this increases the binding affinity. We also included thiazole orange at 2 different positions within the TFO sequence. So one beside the kind of click group and one at the far end. And then we have the DIPCO modification to allow for strain-promoted click chemistry. Initially, we looked at the stability of these TFOs. So we have the DIPCO TFO on its own, also clicked to proteosin and to phenanthroline control. So TFOs 2 and 3 were the most stable in this instance. So that's with the 2 thiazole orange modifications on TFO 3 and 1 thiazole orange modification at the far end from the DIPCO group in this instance. So we looked then at the gel electrophoresis to see the cleavage. So we have the EGFR target and the off target. The EGFR target is depleted while the off target remains undamaged, But it's important that this is in the absence of ascorbate, and these are the first kind of TFO hybrids that don't require a reducing agent to achieve selective DNA damage. And we can compare this then to phenanthroline, which does require ascorbate to achieve the damage, but it's also really poor at forming a triplex in this instance. And we get very little damage to the target, but there's also some damage to the off target. So this kind of shows that the proteosin not only can allow for self-activated cleavage, but it also stabilizes triplex formation. We then quantified this using qPCR, and we can see that at 6 hours, we get about a 70% reduction in the DNA, and at 24 hours, it's almost completely ablated. Finally, then, we wanted to kind of expand this strain-promoted TFO to allow kind of for a screening of large libraries of azide-modified metallodrugs. So in this instance, they're all copper-based. So we developed this self-assembly protocol. So here we incubated the strain-promoted TFO with the target duplex, allowed triplex formation to occur, and then we added in one of these azide-modified groups. The click could be then formed in situ, and then we could add copper or copper and ascorbate, and we got sequence-selective cleavage. We developed an assay then with target and off-target, kind of similar to you've seen before, but we looked at the intensity of the different bands in the gel using band densitometry. And through this equation, then we could plot, kind of give a numerical value to whether it was targeted versus off-target cleavage. We plotted this kind of as a heat map here. So the benzyl DPA in this instance had very poor cleavage activity, which was actually due to kind of destabilization of the triplex, and so we didn't get good triplex formation. Whereas you can see proteosin, which is without ascorbate, has very high cleavage activity, and then it's DPZ, Imfen, and phenanthroline. The final kind of part of my talk today is then I'm going to talk about these monoplatinum DPA derivatives. So these have one labile chloride that combined with DNA, and then they also included in these bulky kind of DPA groups, which are to prevent DNA polymerase read-through. The first step, it's similar to you've seen for the proteosin TFOs, was to look at the stability of these triplexes. Interestingly, in this case, it's actually two different TFOs that were the best. We have TFO3, which is a two thiazole orange modifications, but in this instance, it's TFO1 that formed a stable triplex. And this is where the thiazole orange was in close proximity to the platinum group. This is the opposite for proteosin, where the second thiazole orange was at the end, which was much more beneficial. We looked to make sure they did only form triplexes with their target duplex. And then we further investigated the cross-linking in a similar assay to you've seen before. Again, this is the FAM labeled strand in this time that has had the DNA cross-links with it. And in this experiment, it was really important as it informed the design of our next assay, which was to see could a DNA polymerase read past the platinum adducts that formed. So from the gel electrophoresis, we knew that it was the target strand that the platinum cross-linked with. So we put a 5' phosphate group on the complementary strand. This could then be digested by lambda exonuclease to give single-stranded DNA. Add a primer, it's extended, and we get the duplex again. When the platinum TFO was present, the DNA polymerase cannot read past the platinum adducts, and a truncated product is observed. We looked at this for the range of different hybrids, so TFO1 and 2 with the two different DPA platinum derivatives, but also an oxaliplatin control. Oxaliplatin is much smaller, but it forms adducts with two DNA bases, whereas the DPA platinums are much larger, but only form a single adduct. We can see reduced intensity for all the duplex bands in the presence of the platinum TFOs, and then we could calculate the intensity of these different bands. So the platinum DPA TFOs are more active for TFO1, but for TFO3, we can see less than 20% recovery of the DNA in all cases. Then just to summarize kind of everything I've shown the sequence selective cleavage of AMN TFOs. These can be enzymatically synthesized using SNI PECs. We can develop cis chemotype platinum TFOs to maintain the ability to cross-link with DNA, and that we can achieve self-activated cleavage using the marine alkaloid TFO hybrids. Finally, I showed the self-assembly protocol, which can be used to screen large libraries of these metallodrugs, and also that the platinum DPA TFOs can prevent DNA polymerase read-through. So finally, just to say thank you to everyone in the Kellett Research Group, everyone we've collaborated with on these projects, the funding, and to you all for listening.